thank you for the introduction and thank you for the invitation. It's a pleasure of being here. And I would like to introduce you to something which might be quite novel to many of you, and this is the Human Glycome Project. And what did we learn from the analysis of the first 100,000 people? But before I start, I do have to disclose that I'm a founder of the Genus Glycoscience Research Laboratory, which is a private research institution, but also a couple of other companies, including Glycanage, which I will be mentioning during my talk, and I do have a couple of patents in the field. So we all know that patient stratification is the holy grail of modern medicine. To be able to treat patients properly, we have to first somehow to measure different biomarkers to classify them into the proper diagnosis, into the proper therapy which they will or will not respond. And the majority of people in the last several decades focused on genes and looking at genetic data to stratify patients. But despite the technical success, genetic research did not fulfill our expectations. After hundreds and hundreds of millions of euros spent into genetic research, we have very little clinical translation. There is some translation in the field of pharmacogenomics. There is now a little bit more in the field of oncology, but still we have very little use of genetic information in the majority of complex diseases. And there is a number of reasons for that. And the first one is that genes do not define everything. Actually, only about 30% of complex diseases is genetic, and only about 20% of our longevity is genetic. And the best example of that are the identical twins. When we look at these two pairs of twins, with every single letter in the genome being identical, they still have quite different life fates. And this is because genes do not define everything. And actually, most of our complex diseases are environmentally caused and not genetic. And even the parts which are genetic, it's not easy to link individual change in the DNA sequence with the final phenotype, because individual SNPs very rarely directly cause a phenotype. Most of the time, they have to interact with different SNPs, with the environment, with the epigenetic information, through a number of layers before they come to the final phenotype. So by looking at the DNA sequence, it's very hard to predict the phenotype. And one part which is also very hard to predict from the DNA sequence are the glycans. Glycans are important structural components of nearly all proteins. Practically all proteins, all the membrane, and outside of the cell are glycoproteins. Many intercellular proteins are also glycoproteins. So we have these complex glycan structures attached to different parts of the polypeptide backbone, and the molecule functions as a single unity. So both the peptide and the glycan part determine the structure and the function of a molecule. And when we look at the complexity of different biological molecules, DNA and RNA are built from 4 building blocks. And then proteins have 20 different building blocks. While at the same time, glycoproteins have thousands of different building blocks which can be added to the polypeptide backbone, which strongly increase the biological complexity of life. And glycoproteome is several orders of magnitude more complex than the proteome. And actually, glycans are responsible for the large part of individual variability. So the fact that we're all not the same is by large defined by the glycans. And actually, we all know that. For over 100 years, we know that people have different blood groups. And blood groups are glycan structures. And actually, the first example of successful personalized medicine is a blood transfusion, which is based on analysis of glycans in different people. So to understand inter-individual variability, we have to look into glycans, and we have to include glycans to understand the whole picture that we are looking at. And maybe one of the best examples of that are the monoclonal antibodies. So there are hundreds and hundreds of monoclonal antibodies, either in clinical use or in development, which are wonder drugs of today's medicine. And one of the examples is rituximab. Rituximab is anti-CD20 monoclonal antibody. It's used for over 20 years. and it's killing the CD20 cells. It has only one tiny little problem. The majority of molecules have this core fucose in their structure. And the core fucose is preventing monoclonal antibodies from activating antibody-dependent cellular cytotoxicity. So we are making drug, and 95% of the drug does not work because it has the wrong glycan. So how do we cope with that problem? We give more drug. So we give huge doses of monoclonal antibodies, while many of them actually do not work. And then, a decade later, somebody thinks about it and develops ublituximab, which again is an anti-CD20 monoclonal antibody, just it has a proper glycosylation. It doesn't have a core fucose on its glycans. And this antibody is effective in 100 to 500 times lower doses. So just by looking into glycans, we make a drug which can be 100 times cheaper, 100 times more effective. And this is still not happening, but I think this is not too far away that FDA will simply not approve drugs which are wrongly glycosylated. So what happened to the animal insulins when the human insulin was registered will probably happen to the wrongly glycosylated drugs. They will simply be delisted. And it's not me claiming that. There is a very strong policy document by the US National Academies which said glycans are directly involved in the pathophysiology of every major disease. And we need additional knowledge from glycoscience to realize the goals of personalized medicine and to take advantage of the substantial investments in human genome and proteome research and its impact on human health. And we had a similar documents in Europe in 2006, 2015, and actually we are drafting a new one now. But the problem is, the glycan analysis is very complicated. It takes many chemical steps from a biological sample to the final readout of a complex structure like a glycan. And this is particularly difficult to do in a high throughput mode. So when you do globally, there are only 2 labs in the world which do high throughput glycomics routinely. One is the Leiden University Medical Center, and the other one is my lab in Ghana. There are some attempts in NIBRT and MPI, and now we are also developing a lab in Beijing. So there is definitely a need for high throughput glycomics. And this was one of the ideas behind starting the Human Glycome Project, which was initiated in Dubrovnik in May 2017 on a meeting that I organized, and formally established a year later again in Dubrovnik. And currently, we have over 44 individual projects, over 300 participating researchers, which work together on trying to understand the human glycome. And we have 5 divisions of the project. One is the diversity of the human glycome. The other one is inter-individual variability of the human body fluids, inter-individual variability of the tissue glycome, functional relevance, a regulation of the glycome, and analytical methods for analyzing glycome. So I'm leading the part about inter-individual variability of the human body fluid glycome, and I will mostly be talking about it in the rest of my talk. So far, my lab analyzed over 100,000 people, but not any 100,000 people. 100,000 people from the best phenotype and genotype biobanks in Europe, some of them also in US and China, including the Twins UK with over 12,000 twins, including the Sabre cohort, the 10,000 Wild Dalmatian, Orchids, and so on and so on. And actually, we finished this initial fishing expedition. So we collected a huge pile of fish, which for us is these Excel spreadsheets. And now we are trying to mine the gold out of these data. And actually, in the last couple of years, we are becoming quite successful, and we are enabling glycomics to join the mainstream science because we are starting to publish in the leading clinical journals, including gastroenterology, circulation research, diabetes care. These are our papers in the last 3 years. And these papers are also attracting a lot of media attention. For example, our latest paper about the predictive biomarkers for diabetes and cardiovascular diseases got into 0.3% of the most cited, of most tweeted papers ever. So what is so different in glycans compared to all other omics which are currently being done. First thing is, glycans are not directly encoded in the genome, but they modify protein structure. So in this way, they alter genetic information. So when we think about alternative glycosylation, about adding a different glycan structure to a position on a protein, the consequences are very similar to coding mutation. So you have a change in the glycan, which changes the protein structure. But this is not inherited as a single sequence variation. It's being inherited as a complex trait. This is something we clearly demonstrated 2 years ago in a paper in Nature Chemical Biology, where we analyzed a cohort, collaborative cross-cohort of mice, where Grant Morahan from University of Western Australia started with the 8 founder strains, inbred them for 3 generations, actually interbred them for 3 generations, and then inbred hundreds of strains. And when we analyzed these strains, and each strain is the analogy of a human cousin, so we had clones of cousins, and these individuals, or strains, became very different in the way they glycosylate proteins. So this is the composition of their IgG glycome, which is functionally relevant, which makes those mice very different based on a reshuffling of individual SNPs, actually of hundreds of SNPs in the different regions of the chromosome. And this is so because glycans are encoded in a complex dynamic network of hundreds of genes. There are hundreds of proteins in a biosynthetic pathway, but moreover, there are probably even more proteins which regulate those hundreds of proteins which actually execute glycosylation. So in a series of papers in the last decade, we mapped, at the moment, over 33 of them are published, over 40 in our pipeline of genes which regulate IgG glycosylation. So there is a complex network of genes which makes a decision how the immunoglobulin will be glycosylated, and then they somehow regulate the activity of the executive genes, of the genes which actually perform the glycosylation. And this is not a fixed thing, because a polypeptide is something which is defined for our lifetime. We inherited the gene, we have a specific protein sequence. For glycans, this is something which changes. For example, immunoglobulin glycans, which are determining the effector functions of immunoglobulins, change with age. When we are young, they're mostly anti-inflammatory, they suppress inflammation. As we are getting older, they become more pro-inflammatory, they promote low-grade chronic inflammation. So the analogy I like to make is that polypeptide is something like our body. We cannot get out of our body, we are fixed with it, but we can change our clothing. And if there's winter, we can put something which will protect us. But if we move to a warmer climate, we should have a different type of coating, meaning we can put a different glycosylation of a protein. And occasionally, we make a mistake. So if we put the wrong glycan on a protein, it will be functionally less appropriate. For example, these guys will not survive long the Russian winter. And when we think about a cell membrane, and the photo of a cell membrane is here, these are the cells, and on the top of the cells, there is a thick layer of glycocalyx, of glycans, which is much, much larger than the cell membrane itself. So it's either we have this thick fortification, which protect our cells, or we do not have them. And despite the fact that there is no direct genetic template, glycan composition is between 50 and 70% heritable. We did this on a large collaborative study with Tim Spector and his cohort of twins, where we analyzed monozygotic and dizygotic twins, compared their shared environment and genetic component, and concluded that despite the fact there is no gene for a glycan, it is still largely heritable. But as I mentioned, this is heritable through this complex network of genes which work together to make a biological decision which is then being executed. And some of these genes are known risk factor for diseases. And these genes which regulate then in some way affect the final effector genes. And in a paper published actually last week in Science Advances, we show for a little part of this network there is a gene which is a regulator at the transcription level of the enzyme, which finally does the work. You silence the gene, there is upregulation of the enzyme, and there is a change in glycosylation. So when we took this network of genes and looked what is the pleiotropy with other known traits and diseases, we found there are at least 94 different phenotypes which are pleiotropic with IgG glycosylation. So genes which regulate glycosylation are known risk factors for things like systemic lupus, Crohn's disease, type 1 diabetes, but also HDL, total cholesterol, Parkinson's disease. So this does not mean that in all those diseases, genetic effects are mediated by glycans, but quite probably out of those 94, at least some of them, there is a genetic risk which is mediated by a change in glycosylation, which then affects a disease risk. And for example, in diabetes, there are several levels of evidence suggesting that changes in glycosylation are actually driving development of diabetes. For example, there was a paper in 2011 by Jamie Mars and his team showing that glycosylation of glucose transporters is essential for their proper function, and it can be affected by the change in HNF1 alpha and FOXA2 genes at epigenetic level. Also, there is a line of evidence suggesting that hypocellulation of IgG is actually causing insulin resistance in obese individuals. In our first GWAS study, which we did 10 years ago, identified exactly this HNF1 alpha as the master regulator of plasma protein fucosylation. An interesting thing with HNF1 alpha is that if you have a single defective copy of this gene, you will develop diabetes before the age of 45, and this is one of the monogenic types of diabetes called HNF1 alpha MODY. The problem with HNF1 alpha MODY is that it's generally undiagnosed, and since the therapy is very different, it's a great pity that these people cannot be identified, and they can't be identified because there are no proper biomarkers. And then we showed that actually glycans can be a very good biomarkers to identify patients with this rare type of diabetes. But HNF1 alpha is really rare. It's maybe 0.5% of early onset diabetics, which is not so exciting. But the question is, if a mutation in a gene is causing a diabetes, is epigenetic silencing of the same gene one of the pathways to type 2 diabetes in regular population? And first what we did in a collaboration with Lauc and her team, and Lauc is sitting here in the second row, is we looked at glycosylation in the plasma of people and methylation of HNF1 alpha in the same people, and saw there is an association. But association is not a causation, especially when epigenetics is the question. So what Vladka did, they developed a special epigenetic tool. They modified CRISPRs by attaching different epigenetic modulators and managed to change these few sites for which we found there is an association with the change in glycosylation. And what they demonstrated actually that the change in methylation is causing the change in gene expression and then change in glycosylation. So there is a pathway showing that if there is a change in methylation, there will be change in a phenotype which we associate with diabetes. And then we looked in a small cohort of people who had a type 2 diabetes, and we had to look into pancreatic islets, and this is why the cohort is small. And actually, when you look methylation of HNF1 alpha in the pancreatic islets of people with diabetes, there is a subgroup of patients who is very different, who have HNF1 alpha silenced nearly completely with methylation. There is a subgroup of people who is way off the control range. So there are some indications that epigenetic silencing of HNF1 alpha and the changes of glycosylation caused by this silencing could be a pathway to diabetes. So we looked into our big cohorts. We analyzed 100,000 people. Among those 100,000, there are many people with diabetes. And in the two cohorts, one on Orkney Islands and one in London, we saw that there is some information in glycans which can distinguish people with and without diabetes. Interestingly, on a cohort of incident diabetes, where people were sampled before they developed diabetes, there was also some information. So this was published in 2017, and we used it to persuade the EPI cohort to give us access to their samples. So EPI cohort is collected nearly 30 years ago. We got access to the Potsdam sub-cohort of 27,000 people, and out of those 27,000 people, 800 of them developed diabetes after the initial sampling. So we looked into those 800 people and 2,500 of random samples as a controlled, divided them into the training group and the validation sample, and then when we looked into the validation sample, the glycans were quite predictive. Actually, only glycans plus age were equally predictive of type 2 diabetes as the entire German diabetes risk score. This was published a couple of weeks ago in Diabetes Care. So there is something in glycans which changes before people develop diabetes, which is predictive. So we can look at a healthy person and say, yes, you are at high risk to develop diabetes in the future. But here we have to remember that diabetes is not a single disease. So there is no a single pathway to diabetes. So maybe there is a subtype of diabetes where glycans play a major role, and just maybe we have found this subtype of diabetes. When we looked at people who develop hyperglycemia in acute condition, which is a known risk factor for future development of diabetes, we can nearly perfectly separate them based on their glycans on a baseline level. So maybe we can use glycans to identify people who are on one of the pathways of diabetes. And this was the basis of a diabrisk test, which is one of our huge future spin-outs. We haven't spin out the company yet, but we do have a patent protection for the test, which is visible a couple of years before any other symptom. And it seems to be linked to a subgroup of future diabetes patients, that only a part of diabetic patients have this biomarker, and that these glycans are not only biomarker, but they're active effectors in disease development. And the biggest question here is, can we change those glycans, either by lifestyle or pharmacological intervention, and would this change then decrease the risk of future development of diabetes? And this is where we have a couple of large ongoing clinical trials, and this is really exciting, and I'm looking forward to results of these trials. So in the second part of my talk, I will switch to another problem, and this is aging. As we are getting older, we are becoming more and more interested in aging. While I was young, I was not interested in aging at all. Now it becomes more interesting to do some research on aging. And a friend of mine, David Sinclair from Harvard, just published a book saying why we age and why we don't have to. He thinks that aging is a chronic disease which can be managed pharmacologically. And actually there is a paper now showing that if you do, actually it's a quite aggressive pharmacological interventions, you can decrease your methylation age for a couple of years. And we just published a paper which says even if you start exercising, you can improve your glycan age for a couple of years. So maybe there are really ways we can modify our aging trajectory. Because when we look at expected longevity, for centuries, people were living on average below 40 years. And then in the last century, practically all around the world, this has at least doubled. So in the developed countries, people are now expected to live over 80. And how will it go further? Will it be 120 in the next 100 years? Or will it plateau? This is something we still do not know. But a large study conducted on over 110,000 people showed that a healthy lifestyle can increase your expected lifespan for 10 years, but actually it's expanding the healthy lifestyle. So not that you will be old and then suffer for additional 10 years. Actually, you will have 10 years, additional 10 years of healthy life. Now, for decades, we know that the caloric restriction is working. These two monkeys are on a diet for 20 years. And this one is hungry for 20 years. So he does look better, but do we want to live longer and be hungry? I'm not sure this is the best solution. So we have to find a better solution than just not eating. But it is important because what we know is that the path to disease is by large decided with our lifestyle. And unfortunately, most of us, in particular men, go to the hospital where in a stage when they don't function anymore. When some of the organs fail, and then you have so much pain that you cannot function, then you go to the hospital. But then it's too late, because then the changes are irreversible. You can only treat the symptoms. You cannot revert the cause anymore. So the idea of modern medicine is, let's try to find them very early, give them reprogramming therapy, and then try to heal them. But we should actually go here. This is economically not too attractive, so the big pharma doesn't like this part here. Big pharma likes this part here. You treat sick people with drugs every day for the rest of their life. Reprogramming therapy is picking up because now they're selling it for $1 million a patient, and they say, yes, this is cheap because we are not treating them for the next 40 years. But here, this can be done with a simple lifestyle intervention, or maybe some pharmacological interventions where you can reprogram the molecular pathways which are going in the wrong direction and help people go back to the homeostatic conditions. And we do hope that glycans will be a big help there, because glycans often change before any other symptoms of a disease are visible, and you can measure it, and by measuring it, you can actually motivate yourself to improve. So how can glycan clock be involved in a number of diseases, and why is it so important? So glycan clock, or the glycan age, is based on glycosylation of immunoglobulin G. Immunoglobulin G is a major effector of our immune system, while the effector functions are regulated by glycans. And there are now hundreds and hundreds of publications which clearly show that, for example, if you remove this core fucose, this protein activates ADCC. So this is something what pharma industry is doing now, making glycoengineered antibodies. But also, if you cellulate IgG, it converts from pro-inflammatory molecule into an anti-inflammatory molecule. So it's actually suppressing inflammation. Glycans are circulating IgG, and if you think about it, 99% of circulating IgG do not have antigen to bind to. It's just circulating. It's waiting for antigen to come. This circulating IgG is balancing inflammation on the multiple levels. And what we clearly see is that young people are more anti-inflammatory, they are suppressing inflammation, and in old people, glycans are promoting inflammation. So we published this for the first time 6 years ago when we analyzed 4 cohorts of over 1,000 people, where we learned that glycans actually change quite significantly with age. They change in all 4 cohorts in the same way. Some glycans go up, some glycans go down. And actually only by taking glycans and putting them into a mathematical model, you can predict age of a person plus minus 9 years. Now, predicting age plus minus 9 years is not a big thing. Methylation can predict age plus minus 1 year. But the thing is, what explains the difference? What is the difference between the age you predict based on glycans and your real chronological age? And when we looked, this difference associates with all the known biomarkers of unhealthy life. So you live an unhealthy life, your glycans move into the old age faster, and you have more low-grade chronic inflammation, which glycans are not suppressing. So with this knowledge, we went into the world, asked our friends, give us some samples from the population where you live. So we collected 27 different population of people, at least 100 people in each of them, very wide age span, we fully randomized them across 32 96 well plates, analyzed them, and learned that when you correct for age, glycans are practically on a line with expected lifespan and the human development index of a country. So if you live in undeveloped country where your lifespan is actually shorter, your glycans move to this old age much faster than when you live in much more favorable conditions. We do not know exactly why this happens, but clearly this happens. What we also learned is that when you look people with the different diseases, and these are diseases from inflammatory like lupus, ulcerative colitis, Crohn's disease, also some cancer, type 2 diabetes, the changes we see in these patients go in the same direction as aging. So young people with different types of diseases have glycans, which are IgG glycans, which look like the glycans of old people. And clearly, these glycans are not the biomarkers of a specific disease, because they change in the same way in lupus, ulcerative colitis, even type 2 diabetes. But what we see here is probably one of the molecular pathways which contributes to disease development. And this is this loss of the ability to suppress low-grade chronic inflammation. You have too much inflammation, and this too much inflammation ends up in some of the diseases. I cannot go through all of them. There is a recent review paper from my team citing over 300 references, so if you're interested, have a look. What I will focus in the last couple of minutes of my talk would be cardiovascular disease. Because what we see that glycans associate with hypertension, but they change even in pre-hypertension. So even before people have hypertension, glycans are altered. And there is a paper by Phil Scholl from UT Southwestern suggesting that hypocellulated IgG might be causative for obesity-induced hypertension, and it acts through the Fc gamma receptor 2b. In his subsequent paper, he showed that by feeding mice with a MANAC, which is a precursor for sialic acid, you can actually revert this effect and protect obese mice from hypertension. So we teamed up with him for that paper, and we analyzed people. We couldn't treat people with MANAC, but what we showed that in people, the hypocellulation associates with a higher blood pressure, suggesting that this might be similar to what we are seeing in mice. In the meantime, which is still unpublished, we did a small cohort of people who were fed by MANAC, and apparently in humans it does not work in the same way as in mice. So you cannot just feed people with MANAC and expect IgG cellulation to improve, so people are still different than mice. But what we see in people, we see that these glycans, which are changing with age, also strongly associate with the cardiovascular disease risk score. So they associate with the risk of future cardiovascular events. So again, this is correlation. Can we go a little bit deeper and see whether this actually has something causative in it? And again, we looked to this Potsdam cohort, because out of those 27,000 people, there were also 500 cardiovascular events. And when we looked into those cohorts, again, glycans plus age were as predictive as the AHA score, American Heart Association score for predicting cardiovascular disease. So by measuring glycans, you can actually predict future cardiovascular events. Now the question is, if these glycans are really causative, can we change it? So what can we do to change our glycans? And if we change our glycans, will it decrease the cardiovascular disease risk? So we don't have many people who have been tracked longitudinally for their glycans. And this is me. And I'm horrible. So my glycan age is over 70, while I'm still under 50. And it's still going in a bad direction. Besides what happened here. And what happened here is that I took a long holiday and I was hiking for 10, 15 kilometers a day and socializing with these wild goats. So healthy lifestyle improves your glycans. But it's difficult. When I got back to work, it went back again. And then last year, I decided to lose some weight again, and then I gained it again. So it's, yeah, I'm fighting. So this is me. This is not science. But we also did a large cohort of people in Australia, and it does seem that it's central adiposity, which is driving this pro-inflammatory potential of IgG. So we try to focus on it by looking at physical activity. Yes, physical activity can help. This was published. We have some unpublished data. This was done on 2,000 twins measured in 3 time points, and twins which were losing weight with time were actually aging much slower than the twins which were gaining age with time. So this suggests if you're losing weight, your glycans do move into a better direction. And also we did a cohort of people undergoing bariatric surgery. And if you do a bariatric surgery, you can actually improve a lot. It's a very drastic approach. But we had a person becoming 30 years younger after bariatric surgery within the 6 months. So there is hope that something can be done. But it's not easy, because if you just go and start exercising, you actually get worse. We had a couple of cohort of people just starting to exercise a lot. They get worse. There is a very complex interaction with the microbiome, because gut microbiome is a very important element of health, and it is affecting the plasma glycome. So we had a study where people were doing fecal microbiome transplantation, and this change in the microbiome directly caused, and we know it's causative because it was intervention, they change the glycome of a person into more anti-inflammatory condition. And we know that the diet is a very important element here, because in normal physiological conditions, this is your glycome into your gut endothelium. Evolutionary, our guts were always empty. We were hungry most of the time. And in this situation, our microbiome evolved together with us to feed on our glycome. So we can think about our guts like this coral reef with the different fish which we are actually feeding with our glycans. But in the modern world, we are overeating ourselves. And overeating ourselves with ultra-processed food, which has a lot of different chemicals which are killing bacteria, actually. And by the way, this is Tim Spector's son, who for his book was feeding only on McDonald's for a couple of weeks, which changed his microbiome drastically, and he even claims it's for a lifetime. It hasn't recovered in years from now on. So this situation, where there is always food in your guts, it's more similar to Black Friday shopping. So everybody becomes aggressive, too many people on a small place, too much food, and bacteria start to fight each other. So what can we do? How can I change my glycans or my disease risk with lifestyle interventions? In theory, there are 4 components, and we all know them. It's diet, it's physical activity, it's stress, and it's sleep. But we cannot change all 4 of them in the same time. So for a long time, I was hoping that my problem could be sleep, stress, activity. It did not help. For me personally, it's mostly diet. So for my glycans to improve, I have to look like this hungry monkey. And for me, this seems to be the only way. But for some other people, maybe stress is a key problem, maybe sleep is a key problem. We don't know. And glycans will not tell us the solution. But what glycans can do, glycans can be used to evaluate different attempts, different changes, and then in this way be a motivator. For example, my watch is telling me every hour it's time to stand up for a minute. And then it gives me small stars. And this motivates me because I want to have another star, so I stand up for a minute. So if you measure your glycans and you get some kind of a number and some feedback to your efforts, this could help. And we have this Glycan Age company, which is selling this test. And actually, we work with a number of longevity clinics all around the world, where people are using Glycan Age as an objective insight into the current state. So first, you come in and see, what is my current state? And then you have a quantitative tool to see whether what you are doing is working or not. And at the end, I have to say that what I was showing is highly collaborative. We collaborate with dozens and dozens of institutions all around the world, including the Cornell, with Carsten here and Jan in the New York campus. We are generously funded mostly by European Union. We got over 15 million euro in grants in the last couple of years. I have a large group. It's over 50 people now. This is even actually a smaller group, and they do all the work. I just go around and talk. And as the final take-home message, I would like to encourage you to include glycans in your studies because they will enable you to see the whole picture. I was talking about the CVD events and prediction of CVD, prediction of type 2 diabetes, effects on longevity, quantification of lifestyle interventions. I touched up on interactions with microbiome. There is much more there. There is the resistance to pathogens. There is the prediction of mortality in colorectal cancer I was not mentioning. There is prediction of response to monoclonals, which is one of our focuses of research. There is progression of inflammatory diseases. There is a lot of information of glycans, which can be added to all other multi-omic things which we are doing. And you have Carsten here, who published a couple of prominent papers of doing these things, so I think there is a room to collaborate here. And I thank you for your attention.